It's said that a hen is an egg's way of making more eggs. And the same may be said for every organism that reproduces sexually. From plants to fish, amphibians, reptiles, birds, and mammals. All sexual organisms share a common feature. They transmit genetic information from each parent to their offspring through the processes of meiosis and fertilization. The sex cells of each parent contain a unique genetic code in their chromosomes, which combine to create new and equally unique individuals.
This program is about meiosis and the formation of gametes. You'll follow the actions of the chromosomes as they move through the various stages of this process. Like all processes, disruptions can occur, some of which may be considered beneficial or detrimental to the population. You'll learn how humans apply their understanding of meiosis in the fields of animal husbandry and agriculture, and how ultimately sexual reproduction leads to the biological health and diversity of the species.
So these two are the daughter cells.
Then this would be the mother cell.
Right. And the chromosomes that are in the daughter cells should be identical to what's in the mother cell.
Which they are.
What are you two up to?
We're just using these models to demonstrate the process of mitosis, where one cell divides to produce two identical cells.
Okay. And Shailene, what are some of the characteristics of the daughter cells produced by this process?
Well, the chromosomes in the daughter cell are the same number and the same kind as in the original mother cell.
In this case, the mother cell has 4 chromosomes, and the 2 identical daughter cells have 4 chromosomes also.
Good. So, Mark, would I be correct in saying then that mitosis is a process which helps maintain chromosome number in a species?
Right.
Okay. Let me give you a different situation now. In sexual reproduction, a sperm unites with an egg to produce a single-cell zygote. How can chromosome number in the species be maintained when 2 cells unite to form 1?
The sperm and egg would have to have fewer chromosomes than a normal body cell.
Yes, that's right. They'd have to have exactly half of the genetic makeup, or 23 chromosomes.
So when they combined, the normal number of chromosomes would be maintained.
All right. Every cell in our body has 46 chromosomes, 23 pairs. That's called its diploid, or 2N number. Now, the gametes have 23 chromosomes. They're called haploid, or N.
So the human sperm and egg cell would have 23 chromosomes, or half the diploid, or 2N number.
Right, but not just any 23 chromosomes. One of each of the 23 homologous pairs enter the gamete.
What does homologous mean?
Homologous chromosomes are a pair of like chromosomes found in a diploid cell. Let me use this licorice here to demonstrate. One comes from the egg, one comes from the sperm. Early in meiosis, the two of them line up and then separate to form the gametes.
Meiosis?
Yeah, meiosis. It's called reduction division. It's the process in the body that leads to the formation of gametes. A single diploid cell divides to produce 4 haploid cells. Now, can you think of how this might happen?
If these licorice represent chromosomes, one cell could have 2 chromosomes, and when it divided, we'd have 1 chromosome in each cell.
No, that can't be right.
Why not?
Because we have to produce 4 cells, and we only have 2 here.
I remember in mitosis, the chromosomes duplicate in this process. But what happens if it happens here?
What if the cells divided again, and then we end up with 4 cells, each with 1 chromosome, or half the genetic material we started with?
That's very good.
The number of chromosomes vary among different species. Some ferns have more than 1,000 chromosomes in their diploid cells. Dogs have 78, goldfish have 94. Even an amoeba has 48 chromosomes. As far as biologists can determine, there is no apparent significance between chromosome number and species complexity.
In meiosis, there are two nuclear divisions, followed by division of the cytoplasm, cytokinesis. One of the differences between mitosis and meiosis is that meiosis has two distinct divisions. They're called meiosis 1 and meiosis 2.
But what about the division phases we learned about in mitosis?
They're present in meiosis as well. Now, do you remember them?
Prophase, metaphase.
Anaphase and telophase.
Good. In meiosis, they are repeated. The interphase between meiosis 1 and meiosis 2 is usually very short or sometimes not even present. But prophase, metaphase, anaphase, and telophase are numbered 1 or 2, depending on whether they occur in meiosis 1 or meiosis 2.
Oocytes are produced in follicles within the ovaries. Interestingly, the ovaries of a female fetus already contain all the cells that will eventually mature and become egg cells when the female reaches puberty. Prior to meiosis, the chromosomes have replicated during the S or synthesis stage of interphase, just as they did in mitosis. When the chromosomes become visible at prophase 1, each one consists of two identical sister chromatids, joined by a centromere. The chromosomes now pair up with their homologous counterparts. Homologous chromosomes are pairs of chromosomes that carry genes controlling the same inherited traits at the same positions on the respective chromosomes. One member of each homologous pair is inherited from the mother, and the other comes from the father. These homologous pairs form a tetrad of 4 chromatids. During the pairing up of homologous chromosomes in prophase 1, a chromatid from one chromosome may overlap with a chromatid from its homologue. The point of contact is called the chiasma. It is at the chiasma that one segment of a chromatid is exchanged with a corresponding segment from the second chromatid. This process is called crossing over. Crossing over results in variations of genetic information in the gametes. By the end of prophase 1, the nuclear envelope has deteriorated and spindle fibers have appeared. At metaphase 1, the homologous pairs of chromosomes line up as pairs along the equatorial plane of the cell. The spindle fibers connect the centromeres of the homologues to opposite poles in the cell. During anaphase 1, the homologues separate from each other and are pulled to the opposite poles by spindle fibers. Note that the sister chromatids remain attached to the centromere and move as a single unit to the poles. By telophase 1, the homologues have arrived at their opposite poles. Each pole now has a haploid set of chromosomes, but each chromosome still has 2 chromatids. Cytokinesis usually begins forming 2 daughter cells. At this point, new nuclear envelopes may or may not be formed, and the daughter cells begin preparation for meiosis 2. In meiosis 2, if nuclear membranes were formed, they disappear, and the chromosomes again become visible. A new spindle apparatus appears at right angles to the one formed in meiosis 1. As this stage proceeds to metaphase 2, the chromosomes align on the equatorial plane in single file with their sister chromatids, facing the opposite poles. The spindle fibers again attach to the centromeres. In anaphase 2, the centromeres split, and the sister chromatids separate and are pulled to the opposite poles. The chromatids are now considered chromosomes. With the onset of telophase 2, the spindle fibers disappear and new nuclear membranes are generated around each set of chromosomes. As the nuclei reappear, cytokinesis divides the cytoplasm and 4 haploid daughter cells are formed. This process occurs in all vertebrates.
If meiosis leads to the production of gametes, where in the body would you expect this process to occur?
In the testes in the male.
In the ovaries in the female.
Okay, good. Let's start with the male now. The process of producing sperm in the male is called spermatogenesis. It occurs in the seminiferous tubules of the testes. Here's a cross-section of the seminiferous tubules. Do you recall any of the cells present?
I remember spermatids and sperm cells.
That's a start.
I recall that if you look at the cross-section of the tubules, you'll see the immature cells near the outside and the more mature sperms near the center of the cell.
Okay, now let's see if we can fill in some of the details. Along the wall of the seminiferous tubules are spermatogonial cells. They undergo the process of mitosis. What would that do to the number of cells?
The cell number would increase.
Good. Now, for reasons yet to be determined, some of these spermatogonial cells enlarge and become primary spermatocytes.
And these cells undergo meiosis 1, producing 2 secondary spermatocytes.
The secondary spermatocytes, they undergo meiosis 2, resulting in the production of 4 spermatids. Now, would you expect the spermatids to be haploid or diploid?
Haploid, and would lose most of their cytoplasm and develop into a mature sperm. Notice the flagella?
Good. So, in spermatogenesis, a single diploid cell results in the production of 4 haploid mature sperm cells.
Well, that can't be the same in females.
But why not?
Well, a female only releases 1 egg per menstrual cycle.
Good point.
The production of the egg in the ovary is called oogenesis, and Shailene is right. Only 1 ovum or egg is produced. Let's see if we can model the process here. These are oocytes undergoing meiosis 1. Cytokinesis has just occurred. What observations can you make?
Well, both cells have the same number of chromosomes, but only one is getting all the cytoplasm.
Okay. Now, here's the same cell undergoing meiosis 2. Now what observations can you make?
Well, the same thing happens. One very large cell is produced.
And that very large cell is called the ovum or the egg. Notice that it's haploid. Only 1 chromosome is present as opposed to the 2 at the beginning. And it has the majority of the cytoplasm. The other cells produced are called polar bodies. And they're basically non-functional in the body.
But what about plants? Do they have testes and eggs?
They don't have testes. Moss produce sperm and egg. And flowering plants produce pollen, which develop into sperm nuclei and fertilizes an egg in the ovary. So there are some similarities.
In many organisms, including humans, the production of the egg is different from the production of the sperm. As oogenesis takes place, there is an unequal distribution of the cytoplasm. This leads to the formation of 1 egg cell and 3 cells known as polar bodies, or non-functional eggs. The polar bodies have considerably less cytoplasm than the egg cell. In spermatogenesis, 4 functional sperm cells are formed. These cells may undergo further cytoplasmic development to become mature gametes, as seen in the case of spermatids, which develop tails and become sperm. Haploid gametes unite during fertilization, N plus N equals 2N, to form a diploid zygote. The number of possible chromosome combinations in the gametes is 2 to the N, where 2 represents the number of homologs in a pair, and N equals the haploid chromosome number. In humans, 2 to the N is therefore equal to the possible combinations that each parent contributes. That means that one parent can produce a gamete with over 8 million different combinations of chromosomes. The number of chromosomal combinations in a diploid zygote is 2 to the nth power squared, where squaring represents a union of gametes from 2 different parents, male and female. For humans, this is 2 to the power of 23 squared. A fertilized egg could then contain one of more than 70 trillion genetic combinations. Genetic variability in sexually producing organisms is enormous.
So, I hear you're going to reenact the nuclear events that take place during the process of meiosis.
Yes. We represent a nucleus with a diploid number of 2. Us and our homolog. Our shirts and our caps represent our genetic information.
Now, prior to prophase, we've already been replicated. Now, each chromosome is represented by 2 identical chromatids, and these are held together by a centromere.
In prophase, the homologous chromosomes come together to form a tetrad consisting of 4 chromatids. During prophase, there is crossing over, which is an exchange of genetic information, and we represent this by exchanging our hats.
In metaphase 1, the homologous chromosomes line up on the equatorial plane.
And then in anaphase 1, the chromosomes separate and move to the opposite poles.
In telophase, the two nuclei contain one replicated chromosome each.
And then we move to meiosis 2. In metaphase 2, the chromosomes line up on their equatorial plane.
And then during anaphase 2, the centromere splits and the chromosomes move to the opposite poles.
As a result of telophase 2, we have 4 haploid cells with 1 chromosome in each nucleus.
And each cell contains unique genetic information represented by our shirts and our hats.
Oh, very interesting. Well done, gang.
Dr. Martin, what role do polar bodies play in oogenesis?
Well, remember, oogenesis is the formation of an egg, and a polar body is like a byproduct of that formation. So you need to go through 2 cell divisions, and after each cell division we get one big egg and a little polar body. And so we're increasing the size of the cytoplasm and yet dividing the genetic information in 2.
What causes non-disjunction?
If you remember from cell division, normally we should have all our homologous chromosome pairs pair up and divide nicely into the 2 cells, whether they're sperm or eggs. And what happens with non-disjunction is that the 2 chromosomes pair up and then they don't divide, they both go into 1 cell. So you have an extra chromosome in 1 cell and a missing chromosome in the other cell. We don't really know why it occurs. We know that it's extraordinarily common in humans. For example, of all spontaneous abortions, half of them have chromosome abnormalities, and half of those are caused by non-disjunction. So it's a tremendous problem in humans, and we have some theories of why it occurs, like there hasn't been enough recombination between the chromosomes to hold them together, so there's no longer anything holding them together, so they fall apart and just randomly go into a cell rather than going in an ordered manner. But we're not entirely sure of all the possible reasons.
Suppose these threads here represent the chromosomes lined up at metaphase 1 of meiosis.
Okay. What would happen with non-disjunction?
Well, assuming that anaphase 1 occurs normally, the chromosomes will divide as they should. But when we get to anaphase 2, we'll assume that these chromosomes still separate as they should. But over here, when non-disjunction occurs, these chromosomes fail to separate. So what we end up with are 2 gametes, one with 1 extra chromosome and one with 1 less. Now, if either of these 2 gametes are involved in fertilization, then in most cases the embryo will die and be miscarried.
Okay, that's a little clearer. Good.
One of the biggest outcomes is pregnancy loss, because having a whole extra or missing chromosome is a bad thing for a cell. It can't handle that much extra genetic material or that much missing genetic material. So the biggest outcome is pregnancy loss, and that can be a spontaneous abortion. It can be pregnancy loss even before a woman knows that she's pregnant, so before implantation. It can lead to a stillbirth, and it also can lead a small proportion to live-born babies that have congenital abnormalities and or mental retardation. There are different syndromes depending on what chromosome is extra or missing, and probably the most common syndrome that most people would know about is Down syndrome, which is an extra chromosome 21. Other ones that we see in live-borns are an extra number 13 or number 18, and these are much more severe than Down syndrome. About 90% of these are dead by a year because they have severe congenital malformations. Some of the milder ones include the sex chromosome. So if you have a missing X chromosome, that's Turner syndrome, and these girls are completely mentally normal but have infertility. They're sterile so that they can never have children. And sort of like the opposite of that is Klinefelter syndrome, where a male has an extra chromosome, an extra X chromosome. He's male, but again is sterile. One of the biggest factors that we know to be associated with non-disjunction is increasing mother's age. So, for example, a woman of 20 has a risk of about 1 in 1,500 of Down syndrome, whereas a woman of 40 has a risk of about 1 in 100. So you can see there's a big increased risk there. So one thing that people can do is have children when they're younger. And another thing is there is the possibility of prenatal diagnosis. As a woman ages, we can take a sample of the cells surrounding the baby and tell if the baby has trisomy 21 for Down syndrome or any of the other ones as well.
The condition in which an organism possesses more than 2 complete sets of chromosomes is called polyploidy. If you've noticed that some of the fruits and vegetables in your supermarket are much larger than those found in the wild, they may have been bred as polyploid, with 2 and 3 times the normal chromosome number.
Now, we studied crossing over in meiosis. Shailene, can you tell me exactly what that is?
Well, it would probably be easier to show you. I've used yellow and red yarn to represent a single homologous pair of chromosomes. And as you can see here, before they divide, they replicate. Now I've used marshmallows here to represent their centromeres. The replicated homologs line up next to each other.
So where exactly does the chiasma occur?
The chiasma is right here where the yarns cross over. Now when the chiasma occurs, the matching portions of the chromosomes may be twisted and are exchanged in anaphase 1. During metaphase 2, they realign, and if I can have your help for this next part, in anaphase 2, they separate once more.
So now there are 4 different types of chromosomes, where there were 2 in the first place.
That's right. There's a yellow one and a red one, and as a result of crossing over, there's a yellow chromosome with a red segment and a red chromosome with a yellow segment. Because of this increases the chance of variability in the offspring.
Can there be more than 1 crossover for each chromosome?
Yes, there can. And this really reshuffles the genetic information contained on the chromosome.
Crossing over is a normal thing that happens in cells, and what the cell is doing is increasing the variability. So if we cross over from one chromosome to another, we're sort of like reshuffling cards, and the individuals, the next generation, have more variation. So it's a very important part of meiosis, and it seems the most important thing that initiates crossing over is double-strand breaks in the DNA. There are special recombination enzymes that cause these double-strand breaks.
Is there any normal rate for crossing over?
Yes, we know that for individual chromosomes, they have their average number of crossovers. So for a big chromosome like number 1, we expect 3 or 4 crossovers, and for a teensy one like chromosome 21, we only expect 1 on average.
Well, thanks, Shailene. Now, we've been studying meiosis, and meiosis is associated with the process of sexual reproduction. What are some of the advantages of sexual reproduction?
There's variety in a species.
What do you mean by that?
Meiosis creates sex cells with a random assortment of chromosomes. When they combine, there's potential for variety.
And when you look at the crossing over that occurs during meiosis, the variety is even greater.
Practically all complex organisms reproduce sexually, and therefore are able to evolve through natural selection.
Good. Natural selection is the differential selection of traits best suited for survival of a species. Those traits that are selected tend to spread throughout the population. The most successful combinations of variations tend to become more frequent to the population. The less successful ones become less frequent.
So that's what they mean by survival of the fittest.
Exactly. It's sexual reproduction that increases the rate at which these genetic combinations are created. If it was left to mutations alone, the process of evolution would be very slow. Few mutations are beneficial to the population.
Could you review mutations?
A mutation is a change in the genetic material. This licorice here represents a chromosome, and the jelly beans, the genes on the chromosome, and a mutation is a change in the genetic material. Mutations can occur in body or somatic cells, or they can occur in gametes. If they occur in a gamete that's involved in fertilization, then that mutation gets passed on to the next generation.
What causes the mutation?
There are various causes. Mutations can be caused by radiation, chemicals, or sometimes a mistaken replication during interphase before meiosis. No matter what the cause, any change in genetic material is called a mutation.
Animal breeders, on the other hand, face another problem. Given a small number of possible mating pairs within a population, the same two N combinations are exchanged over and over, leading to inbreeding. Along with this comes a host of problems linked to an increase in the frequency of harmful traits carried by the parent's chromosomes. This also occurs in some wild populations. Wild cheetahs, for example, have so little variability in their gene pool that the skin of one cheetah, if transplanted to the body of another, will not be rejected. Their immune systems can't distinguish between individuals. This lack of diversity in the gene pool is a result of the declining cheetah population.
Triple champion Labrador means it's a dog that a lab that has its championship in 3 different areas of competition: shows, field trials, and obedience trials. I love the Labrador Retriever breed and its versatility, and so when I'm breeding, I want to try and either improve or maintain the qualities that I like in the dog, which is their looks and their temperaments and their working ability, and also we try and work on eliminating hereditary health problems. With labs, the ones that I've run across anyways, there's hip and elbow dysplasia, epileptic seizures, there's 4 or 5 or 6 different eye problems that are hereditary, and there's probably lots of other ones too, but those are kind of the main ones. When you're trying to stay away from a hereditary fault, you try and find out if the parents have it or not. Sometimes that's easy to do, and sometimes it's not. First of all, you wouldn't breed to a dog that had a fault like that for sure. The other thing is they can be carriers. So if it's a known carrier and has produced a certain fault that you're trying to stay away from, that'd be another reason you wouldn't want to breed to that dog or breed from a dog. I'm hoping that maybe someday we'll be able to use DNA testing to find out carriers for certain faults that we wouldn't otherwise know about. Some faults don't show up in a dog, especially some of the eye problems or seizure problems don't show up till they're several years old, and they've already been bred. So if we could find out right away with the DNA testing, it would help in eliminating some of the problems.
We still haven't answered the perennial question of what came first, the chicken or the egg. But we have learned something about how and why it happens. It begins with meiosis. A sperm unites with the egg at fertilization, forming the first cell of a new and unique individual, helping to strengthen and diversify the species.